Meredith Turnbow, we meet again. How are you?
Doing well. How are you?
I'm doing so well. The last time we were together, we were doing a deep dive into DNA, what that means in terms of criminal investigations. We touched a little bit of everything. We went really wide, but not as deep as I think we could have gotten. So here we are again to really pick apart some of those topics, which is so exciting for me as a true crime podcaster, as an investigative journalist. I'm definitely not an expert in the things that you're an expert in, but I am an expert in being a student, I think. So I'm taking the seat of student. I'm going to lead us along this first topic in this series, the start to finish process from collecting DNA to actually getting meaningful results, or let's just say results, period. And I think in the true crime space, or even in, the dramatized TV version of solving crimes, we have this very truncated, express view of how this all happens. So hopefully you'll be able to expand that for us. I know you will be able to expand that for us. So we have a greater understanding of what it actually takes to learn information and get results from DNA at a crime scene. So let's start. Let's start at the very
All
beginning. A
right.
very good place to start. How
Sure.
are we collecting evidence at a crime scene? What does that very first step in the process look like?
Yeah. So, I mean, I'm glad you mentioned TV because I think this is one of the areas where things differ. Um, I think on a television show, the main character does everything. So they're doing the crime, they're working the crime scene, they're chasing suspects, they're working in the lab. There's really just not that many hours in a person's day, and so those are typically different people. Um, So depending on the jurisdiction, the crime scene may be worked by dedicated crime scene technicians. If it's a little bit smaller of a place, then sometimes lab people do work the crime scene. The people working crime scene may be sworn officers that have special training for this. So that's a kind of a good place to start. How evidence is collected is going to depend on the scene itself. So there are some, if we're talking about biological evidence, which when we're going for DNA really is key, there are a couple ways that you can visualize things at the crime scene to help you narrow it down. I think one thing that TV gets wrong, blood is not red for very long. Blood mostly turns kind of a rusty color. It's sort of brown. And so it's not as obvious as it would be on television with bright red kind of paint looking stuff. And so sometimes you need to be able to visualize not only blood, but other biological materials. So you can do a couple cool things on site at the crime scene. You can use an alternate light source. And so instead of just looking at our visual spectrum of light, we can look outside of that into ultraviolet and infrared and use special flashlight that shines those different wavelengths. Things like saliva, urine, and semen will often glow under ultraviolet light. So next time you check into a hotel, you may or may
Oh
not want to
no.
look with one of those first. This depends on how much you want to know. Um, might at least check the bedspread.
I don't know.
Uh,
Ignorance is bliss in that
yeah,
scenario, I
right.
think.
Or you may just put it out of your mind and be like, this place has 4 stars and it's great. Um, blood, on the other hand, it does the opposite of glowing. It'll actually absorb light, and so it'll be like a dark spot under these alternate light sources. Now, if you wanted blood to glow, which they often do for TV, there is a thing called luminol that you can spray, and especially on aged blood stains, it works better if they're kind of dried as opposed to fresh. Then blood will glow with luminol. You need to have it pretty dark, but you can spray that on there and then visualize blood stains that way. So that helps when you're looking at the crime scene as a whole. Like, what do I take back to the lab? Depending on the lab and how big it is and what their evidence receiving area looks like, you may not be able to take everything that you want. And obviously you're not going to roll up carpet from an entire apartment and take that whole thing back. You're going to want to take a cutting, places that are the most interesting or the most likely to have biological specimens on them. You know, you may or may not take an entire comforter back. You might, but you might also take cuttings. It just sort of depends on their protocol. So we can kind of look at the crime scene and see what's there, try to anyway, and see what's there and see what the best items to take back. Now, there are times, say a crime is committed in a vehicle. There are places where they have entire vehicle bays, you can tow the whole car back to the lab and they can work it like that. But not every place has that ability. So again, you may be identifying, kind of narrowing down the places that you want to take cuttings from and then take pieces back to the lab. So evidence is going to be collected like that. You want to keep things dry. And so plastic packaging is kind of a no. You're going to want to package things mostly in paper bags. That way they can breathe. Because if you take, as we all know from refrigerator experiences, if you take your vegetables from the store in their little plastic bag and you put it in the vegetable crisper, also known as veggie hospice, and you leave them too long, they will mold and go bad. Same thing will happen with, say, a blood stain. If it's still damp, you put it into a plastic, it's not going to be able to dry out. It's going to mold, and then those microorganisms will actually eat the DNA. So that's not good. So you want things to stay dry. So mostly paper, paper bags. There's plenty of paper evidence bags, and you want things to have, you know, to be properly labeled. Obviously, where did this come from? Who put it in there? Make sure it's taped shut with evidence tape so that we know that this hasn't been tampered with, that it was collected properly. We know who collected it, we know where, we know when, and then that gets transported to the lab. For things that are occasionally for things that are not DNA, like if you're looking for accelerants in an arson case, they'll put stuff like that into like a paint can, and that'll trap that volatile compound, and then they can do chemical analysis on it later. Since we're mostly talking about DNA, in general, you're gonna have things in bags. There are gonna be things that you're not gonna actually collect the whole item. You might swab it, and so you take basically it's like kind of a lab grade Q-tip, and depending on what it is, you might have a sterile solution that you would wet swab, and then a dry Q-tip that you would dry swab, or, you know, depending if the blood is still wet, then obviously you don't need the wet swab first. But, and you know, this will kind of depend also on the labs or the crime scene tech's SOP or training. But you can swab things and then just take the swabs back to the lab, and those are more convenient to be processed later, just sort of depending on what they are. So touch DNA, or somebody's cut themselves on a piece of glass when they were breaking into a window, and you get the blood off of there, those are going to be things that you would swab instead of take back. And those are usually have their own little container that you collect that stuff in. So that was a lot. Does that make sense?
Well, there's a lot.
Did I leave anything out?
No, I mean, there's a lot to consider at a crime scene, and it's really, I mean, a case by case basis. You know, you're hoping the investigators on scene, you know, kind of have some foresight into what could be important, even if it might potentially appear innocuous at first glance. And what I've been learning and, you know, looking at so many of these cases, that there's so many places along the way of evidence collection and, you know, biological sample collection, all of that, where things can go wrong or it can be degraded or something can happen. And one of the concepts I see brought up all the time, and typically it's in a defense argument, is chain of custody. So can you talk a little bit about that? Why is that so critical to DNA testing as far as, you know, criminal investigations goes?
Yeah, very important. And this was one of the things that they really hammered down on during the O.J. Simpson trial, the first one for the murder of his wife and their friend. So chain of custody is exactly that. If I'm the evidence tech, I collect it at the scene. I have I've got my initials on it. It's taped shut. We're going to then transfer it to the next person. We're not just leaving this stuff in a pile. That's going to get transferred to the lab. At evidence receiving, they're going to say, okay, yes, I have received this from you. There will be paperwork that was received. We know who received it. We know when, and then it will be kept in a locked facility until the lab is ready to work on it. And then when the lab comes to check out evidence, the person from the lab is going to say, yes, this is me. This is when I checked it out. And then anything that was done to that evidence. And so we have to have that chain of custody, because otherwise, if I just left a brown bag of evidence out, I mean, anything could have happened in the, kind of in the intervening time. And so if evidence is mishandled and you break that chain of custody, we lose confidence that that evidence is what they say it was, and it hasn't been changed or altered or mishandled in any way. So yeah, chain of custody is huge. Definitely want to make sure that you maintain it, and that whoever's receiving that evidence is the person that's supposed to be receiving it. You can't just drop stuff with a random person that you see in the hallway and be like, hey, take this to the lab for me. It really needs to go to evidence receiving, that kind of thing. I think people have gotten a lot better about that, because in a lot of times of defense saying, hey, well, this was mishandled. We know that if you break the chain of custody, that evidence is going to lose the confidence of the jury or whoever's looking at it.
Yeah, or could not be used at all, which can really be detrimental, to bringing a case to conclusion. So let's go through this timeline with the best case scenario, that everything chain of custody has been followed. It makes it to the lab. Knowing that there is a whole mess of different types of evidence you're receiving, what would be an initial step taken when that DNA evidence, when biological evidence, when anything arrives to the lab? What's the first step that it goes through?
So generally speaking, the first step, most places, is going to be what's called serology. And so we're looking for biological fluids. And that's, that's if we've got, say, bedding. We're not going to cram an entire king size bed sheet into a tube and try and extract DNA from it. We need to find the places on the bed sheet that are most probative, that we think have the biological evidence that's important. So there are two kinds of serology tests. There are presumptive tests, and then there are confirmatory tests. And so I talked about luminol, spraying the blood and then it glows. That's a presumptive test. We can presume that there is blood there, but we know that luminol cross-reacts with other things. Certain metals, you got to be careful around doorknobs and things like that might be brass. And so that's a presumptive test. Confirmatory tests, before DNA was done, confirmatory tests were much more important, because that was all you had. You said, we can confirm this is human blood. And so there are like Hemotrace sticks would be a confirmatory test for human blood. And so these don't cross-react with as many things. I think they will test positive for ferret blood. Fun fact. So, but generally they are specific for human hemoglobin. And so that would be a confirmatory test. So presumptive would be like it glowed with luminol. Confirmatory would say, okay, we did a Hemotrace stick and it reacted for the presence of human blood. Serology is practiced to greater and lesser degrees depending on the agency. Like I said, it used to be super critical, because that would be kind of the only way you could say we have human blood. Now, with DNA testing being more prevalent, I think some of that has fallen away, but we still do a lot of serology. And so there are presumptive and confirmatory tests for semen, for blood, things like that. So they'll do serology, and basically you're trying to narrow down, what am I looking at? And also to take photographic evidence and say, okay, these are the spots, this is where things were, especially if you have an item that the whole thing is brought into the lab. you'll want to image that and preserve those images as part of the evidence handling process. And so, because the next thing you're going to do is destroy part of the evidence to get DNA out.
Say more about
And
that.
this, yeah. So if we're back to the bed sheet, you know, say maybe we did presumptive and confirmatory testing to say that this is human blood on this sheet. Now we're going to take a little piece and we're going to cut it out. And that little piece is going to go into a buffer that's going to separate the cells from the bed sheet so that we can break those cells open and get the DNA out of them. And so that process of separating the DNA from everything else, so the inanimate bed sheet, the blood cells themselves, any dirt, any of that kind of stuff, but just separating the DNA out from that is called DNA extraction. And so we're going to extract the DNA from the evidence item. Now we had to cut up a piece of sheet, and that is now, that little square that we used is going to be more or less devoid of biological evidence at the end of that. Some places save the cuttings afterwards, but not many, because the whole goal was to get all the cells off of it. So yeah, so that would be the first step. So we visualized it, we preserved any visuals that we thought were important. Now we're going to take a piece, we're going to cut it out, or if it's a swab, we'll use maybe part of that swab, half the swab. If we took 2 swabs, we might use a whole one. It sort of depends. We'll get the DNA out of that, get that DNA clean. And so that's, you know, process 1, 2, and 3, kind of, where we preserve the evidence visually. We examined it for biological fluids, or maybe vice versa. We took cuttings, or decided what piece we were going to use for DNA extraction, and then we did the DNA extraction. So now we have clean DNA, we hope. Fingers crossed. So DNA extraction is really the first step where we're really just working with the DNA instead of a piece of evidence. So now we've got DNA in a tube suspended in liquid. The first thing we're going to do, we want to make sure that there's DNA there, and that there's human DNA there, not just any DNA. Could have been, you know, your dog might have broken a toenail. Now, if we did confirmatory testing, we would probably know that that was not human blood at that point. But if we skipped that, or that lab didn't do that as a matter of course, we might not know. So we're going to quantify the DNA is the next step. So we've extracted it, now it's clean. Now we're going to quantify it. How much DNA we have. And the modern DNA quantification tests are very specific for human DNA. And they have locations that they're going to amplify that are on autosomal chromosomes, so chromosomes that everybody has, and on the Y chromosome, so that we'll know if we have male DNA. And so the quantification is going to tell us how much. It'll tell us if there is Y chromosome DNA present. And then the modern kits also have what's called a degradation index. So as DNA sits, it degrades. And part of that degradation is it gets chopped into smaller and smaller pieces. So degradation index is going to work by, we're going to amplify a really big piece, and then we're going to amplify a small piece, and then we're going to compare them. So if we have perfect pristine DNA and we had 200 cells, whatever, we would expect to have about the same amount of big piece and small piece. As the DNA degrades, we're going to have less and less big piece, because those parts are being chopped up into the DNA itself is being chopped up into pieces. And so we're not going to successfully make a copy of this whole big long piece, but we will still make a copy of this little piece. So we can compare those two, how much we got from big piece and small piece. And the more the big piece falls out, the more degraded that DNA is. So we can get a degradation index. Also, we're going to look at the quantity of DNA that comes from cells that everybody has, or from chromosomes that everybody has, versus just the Y chromosome. And that's going to give us a ratio. So we've got a ratio of human to male DNA. So if we have a female victim, we might have a lot of her background DNA, and a male suspect, we might have a smaller amount of his DNA. And that's going to inform us as to how we go forward with that. So we can learn a lot just from quantification. And what's nice is you extract the DNA and maybe you have that in 50 microliters, which in the bottom of a tube is going to be about this big. That deep anyway. Like this. Not a lot, but it's a lot for forensics. And DNA quantification usually only takes 2 of that 50. And so that's a super informative step that doesn't use much evidence. So that's going to be kind of your informative step. Now you're going to decide, what do we do? So if I've got plenty of DNA, doesn't look degraded, it looks like the human and male quantities are the same, then we can treat this maybe more likely as a single source and go forward, or at least it's multiple males, and then we could go forward with normal autosomal STR testing. If, say, the male DNA was super high, or backwards. If the human DNA was super high and the male was super low, we're not going to be confident that we can get that male autosomal, auto means self, with that male STR profile, because we're probably mostly going to get the background female DNA. And so we could choose then to maybe just look at the Y chromosome, because we can amplify that separately. This is probably way more information than you wanted.
No,
but
not at
quantification
all.
is super important. And that's where you make your decisions in your flowchart. Like, okay, if I get this, then I do this. If I get that, then I do that. If I get no DNA, often we'll stop and say, hey, there's nothing there. It doesn't make any sense to go forward. So there are a lot of decisions that can be made just off of quantification.
Let me ask you this, along the way, sure, what's the interaction with law enforcement or the detectives, do they play a role in deciding what steps are taken next, or is that left up to the expertise of the people in the lab?
In general, those decisions, once the evidence comes in that the crime scene or the detectives want tested, once that evidence comes in, it's mostly going to be in the hands of the lab. The lab will reach back out for things like, remember I was talking about, you might take half a swab or a small cutting. If the lab says, hey, we need to use this entire sample to consume all the evidence to get enough DNA to do testing, they'll often reach back out and say, hey, do you want us to do that? Or do you want us to wait and save that evidence in case we wanted to do something else with it later? New technology or something like that. They will often ask permission to consume evidence. But other than that, when you get to the cutting, the extraction, the quantification, and then where you go after that is often going to be the lab making that decision. Okay. That's a good question.
So then you have the DNA, and you said that you can learn a lot from it, from these early steps, but it's my understanding that there's a lot of translation and comparison, and there's a lot of work that goes into, or steps that go into actually learning. It's not like you have DNA and it points to a person kind of thing. That's where things get condensed in a lot of storytelling about crime scene investigation or criminal investigation. So can you unpack that a little bit? Like, let's do it.
You mean in normal life, you don't like put a bloody swab into a machine and it pops up with like their GPS location, their driver's license picture.
Right. And I think we know that, I think we know that, but it's easier to just, it's easier for our minds to kind of put it in that box as, this is done by a fancy computer.
Yes. Well, and if you're trying to fit everything into a 30-minute episode minus commercials, you right, gotta squeeze it down. So yeah, these processes all take longer than they do on TV. Mm-hmm. And the results that they generate are good, but they are very specific. They only tell you what they tell you. They're not, you're not getting all of that other stuff like you would, you know, from a crime scene drama. Yeah, quantification is just going to say yes, there's DNA, no, there's not. If there is, how much, and what kind of shape is it in, and is it, you know, is the male to human ratio, 1-to-1,
Right.
or is it skewed where maybe we have more than one person?
Mm-hmm.
So that's all you get from there. And then you decide then, okay, now what do we want to do? So if I've got enough DNA that I think I can get an STR profile. So STR is short tandem repeat. So everybody has these repeat regions in their DNA. The difference is how many repeats. And so it's like if you're counting the number of cars in a train. And so everybody has a train, but we're going to count the number of cars, and you're going to have two different numbers for your autosomal. You're just everybody human chromosomes, your non-sex chromosomes. And so I'll have maybe at one location, I'll have maybe 8 repeats from mom and 10 repeats from dad. And so my STR profile at that locus is just singular for the location in the genome that we're looking at, I will be an 810. And then that's it. So we'll look at now, used to be 13 of these locations. Now we're looking at more like 24. So we'll look at these 20 to 24 locations, and we'll get the STR profile. And that's the data that's in the FBI's National DNA Index System. They're the FBI's DNA database. It's also what's in Interpol's database. So there are millions and millions of these STR profiles already in databases, and they're very readily comparable. But you were talking about like interpretation and what does this really tell us? And so that's, I think, where another big deviation between life and TV comes. Because if you have plenty of DNA and it's just one person, then an STR profile is pretty easy to read. You'll see these peaks at an 8 and a 10, and that's me, and that's more or less all that's in there at that location. And then you'll just look at all the 24 different ones and then see what my profile is. But when things get weird or things get difficult, or when you don't have very much DNA, if you're pushing the lower limit of that technology to barely be able to get an STR profile, you might only get a handful of locations instead of 24. You might not get enough locations to reliably compare it against other people because you're going to get a lot of false positives where it looks like they match, but they only matched at 4 places. That does not a whole profile make. The other places where those STR results can get hard to interpret is if there's more than one person in there. And so if I'm an 810 and you're a 7-8 and we're together in the same tube at that location, you're going to see 7, 8, 10. Now there'll be an 8 from you and an 8 from me, so that may be taller, should be taller. So you have a small 8, small 10, and a tall 7 because we share that allele. And with just 2 people, and if you've got a pretty good ratio, it's not that bad. But imagine now you've got 4 people. Now think about all the overlap. And so the gnarlier the mixture is, the harder that is to interpret. And you can't ever say from that kind of technology, you can't say this came from this person for sure. The peaks don't have name tags on them. They're just there. And so, yeah, it gets the interpretation is, it can be very straightforward, but when we're talking about mixtures, when we're talking about degradation, when we're talking about really low quantities, we may not get enough information to be able to make a good judgment on whether this belongs to a person or not, or whether that person is included or excluded. So Yeah, that DNA interpretation part is very frustrating, I think, for a lot of people. The FBI's quality assurance standards require a lot of, or have a lot of rules, especially around interpretation. This is actually pretty strict kind of set of regulations, because when we were proving DNA at the very beginning, this was the only kind of forensic science that you couldn't really see with the naked eye.
Mhm.
You know, if you're talking about firearms or tool marks or ballistics or whatever, that's a lot easier to explain than, hey, I have clear stuff in a clear tube full of clear liquid that I added my magic juice to, and now I say this is a DNA profile. So we have to have really clear rules. And sometimes I think people see the actual output of the instrumentation and there are little bitty peaks down low, and you're like, yeah, I can see it, it's right there. Well, you have to have thresholds. There have to be interpretation guidelines, and you have to say, below this level, we're not confident that this is real versus just noise of the system.
Mm.
And it's frustrating because you're like, I think I can see it, but I think I can see it is not an answer. You can't go outside of your interpretation guidelines to think you can see it, because that's how we end up with bias, where you're like, oh, the suspect's profile is this. I think I can see him in there, even though it's below my
Mhm.
threshold. Yeah, you want to keep those kind of things separate. And so I'm not saying it never happens, but bias is very real. And we do everything we can to minimize that and to have really good interpretation guidelines where we have hard set thresholds and we say, okay, above here it's real, below here, nope. You know, if we have a mixture with this many people or more, we can't interpret it because it's too convoluted. Those kinds of things. And so I think that's probably very frustrating for people. Well, it's frustrating for us too, you know, for people in the lab, but it's probably even much more frustrating for folks that are trying to get answers and the lab says this is inconclusive.
Mhm.
I think
Yeah, that word.
that's hard. And so, I mean, the lab writes interpretation guidelines and they have to follow those all the time. They can't choose and say, no, we're going to follow them here, but not there. They have to follow those guidelines all the time because that's part of the scientific method. That's how we keep from fooling ourselves and talking ourselves into things being there that may not really be there, or explaining things away that really do exist that are just inconvenient to our hypothesis. And so that, I think that exclusion versus inclusion is pretty tough whenever things get weird, which in forensics they do. I think that's kind of the nature of it. But yeah, so that was super long-winded. But yeah, for inclusion and exclusion, I think there are times when you can say this person is absolutely in this mixture. If it's you and me, and you know my profile, we can look at you and be like, man, Kylie's included. She was there.
I
But
hope I'm not included.
and yeah, she was absolutely there. But sometimes it's not possible. I don't know. What else do you want to know about that? I feel like I rambled
Yeah.
forever.
No, I want to know, so can it happen fast? Can the testing, the identification of a profile, interpreting that profile, can it happen fast, or is it always months, years long, which is what it seems like in a lot of these cases that I've been looking at?
So, truth of the matter is, if you had a lab set up, ready to rock, fully staffed, everything in place, ready to go, and they had nothing else going on, and one case came in, I have seen labs turn around DNA evidence same day. There was an attempted child abduction. Guy tried to grab a kiddo through a window, cut himself. Kiddo fought him off, made enough noise. Guy ran away. This kid was asleep at home. The police picked up somebody that someone else had reported as being suspicious. And they said, hey, we can only hold him for so long. Can you tell us if his profile matches the profile from the window? And so, especially when they're just looking at a suspect profile, which is a lot easier to do, they mid-morning, like they had it done because they dropped everything else and just did that because they didn't want to let this guy go. And so it was the same person. They had already worked.
Wow.
They turned around both the crime scene sample and that suspect sample in that case, but the suspects in particular, they turned around very quickly. So if you don't have anything else going on, serology and looking at the evidence, like that can take a while, depends on how complicated the item is. But if you just had a swab and it's ready to go, the DNA extraction should take, that's probably half a day. The quantification, it's gonna be probably 3 hours, setup and everything included. Get that quantification data, and then you want to do an STR profile, depending on your level of robotics and other things. You can get an STR profile in a day. And so if there was nothing else going on, you could turn around DNA results in a couple of days. That's not the problem usually.
Right.
The problem is not how long does it take me to ride Splash Mountain. I'm probably only on Splash Mountain for 5 minutes to 10, maybe. I don't know how long it takes anymore. But the line is 2 miles back. And so unless you have the whole flash pass and you can cut in front of everybody, it's not the process itself that's slow.
Right.
It's the thousands of cases that are in front of you in line. That's what's slow. I've spent a lot of my career trying to help people streamline their workflows, to add robotics, to make things easier, make the dumb things automated so that the smart things that you need your thinking brain for analysis of these DNA profiles and for decision making, that frees up a human to do that. I think that helps a lot. Funding varies a lot from lab to lab. And so how much of this they're able to afford, if your whole lab is just 2 people, it's tough. But some labs are extremely large and very well automated and, run like sewing machine, just all the time. But there's always going to be somebody in front of you in line. And that's usually what's taken a long time, unfortunately.
Right. So we've gone through that process, kind of demystified, what might hold up the process. It did. It absolutely made sense. And now I would love to chat about the lovely acronym we know as CODIS. Can we demystify what that is? It's not the only database, and do the databases talk to each other, and how much interaction does someone in the lab have with the database? What does it actually look like putting a profile on there? Is it like uploading an Instagram photo? Like, what would we say they put it into CODIS and they got a hit? What happens in that sentence?
What does this even mean? Yeah, so I am very guilty of this. I will refer to that whole system as CODIS. CODIS is CODIS is the Combined DNA Index System. CODIS is a software. It's like saying Microsoft Excel. CODIS is a software. In the United States, there are levels of database. There's ELDIS, which is local index system, ESTIS, state index system, and ENDIS, which is the national DNA index system. And so only certain labs have access to CODIS. You have to be a CODIS lab. So there are no private labs in the United States that have direct access to CODIS.
Oh, interesting.
So only the government labs have access to CODIS, and there are very specific rules that you and guidelines that you have to meet to have access. Generally, it's not anybody in the lab putting that stuff into CODIS. You'll have a CODIS administrator, and that is the person that's going to interface with this kind of computer system. They will upload profiles. There's less data in CODIS, or we'll just refer to the whole thing as CODIS, it's fine. There's less data in CODIS than you would think. I was telling you about the number of railroad cars, so if we've got D2, it's a location, we've got an 810, whatever. That profile is going to go in along with the submitting agency and their case ID number, and maybe a couple other things, but that's kind of it. And so if I put a profile in CODIS and it gets all the way up to ENDIS and we get a hit and it's to another crime scene, which happens. Now I can look at that case number and say, I will then need to reach out to that other agency to have them confirm that this is really a match or really, really a hit. If it's to a known individual, say from a state that had arrestee testing or convicted offender testing or something like that, then we're going to know that's that individual as opposed to this just matched to a crime scene in another state kind of a thing. But there will still be a confirmatory process. And so I've got my DNA profile from my evidence. It hits to somebody that was convicted in Kansas. I'll reach out to Kansas and I say, hey, I've got this profile from a piece of evidence. It hit to this guy in CODIS that you put in. Can you confirm? And so they will go back and confirm that person's profile from their submission stuff does match your piece of evidence. And so there'll be a confirmatory process. And then that all gets reported out to the investigator that's working on the case. And so it is not like putting an Instagram photo on Instagram. It is definitely more involved. Every lab I've been into, the CODIS computer is like locked in a room by itself. Only the CODIS administrator has access to that. It's usually keyed or key carded. It's under fairly heavy security because obviously you don't want people putting random stuff into CODIS. That would be bad. And certain profiles aren't allowed into CODIS. So, if I got a person's DNA profile off of their own car, like their own steering wheel, you cannot put that as a crime scene profile into CODIS. It doesn't match it doesn't meet the criteria. It has to be something that's probative. We expect that guy's DNA to be in his own car,
right.
Right. So yeah. So that's different for sure than, hey, I just sat down at my desk and just go, bing, it's a match, or we got a hit. A lot of times those things don't get, they'll process overnight and you'll see them the next day, that kind of a thing. It's not as fast as it looks.
Interesting.
Locked down.
Yeah. So then, have I missed anything in the process of, when a sample gets to the lab or when evidence gets to a lab? Is there anything that I've missed that you want to make sure is included before we get to suspect identified when we go to court on this evidence or this DNA profile?
I think we got it. We want to make sure we're looking for biological evidence if we're going forward for DNA. I would say the only thing that I definitely did leave out would be you might have heard about differential wash. And so if there's a DNA extraction, we're breaking the cells open to let all the DNA out. If we've got, say, like a sexual assault kit, we're going to have a lot of the victim's DNA for sure on any swab, like an intimate swab. But we're looking for sperm, right? or semen at least, but sperm would be super handy. Sperm heads are real dense, and epithelial cells are kind of like big and floaty. And so if we want to separate sperm cells from epithelial cells before we go to DNA extraction, what we can do is we can suspend all that stuff in a buffer that won't break open sperm cells, and then centrifuge it down. So we'll spin it real hard. I don't know if you guys, they probably outlawed these, but like we used to have the Gravitron at the amusement park, where you
Oh,
stand
yeah.
up against the wall and it spins real fast and you stick to it. And not everybody sticks, right? If you're too light, you'll slide down the wall. So it's kind of like that. So the sperm are real heavy and they get slung towards the bottom of the tube. And then the epithelial cells are kind of broken open and floaty and they stay up in the solution. And so we'll pull that stuff off, wash the sperm cells, do it again 3 or 4 times, and then we can have a pellet of sperm that then we can put in a different solution to break those open, and then we can get DNA that's basically just from the sperm without so much of the contaminating background DNA from the victim. So I skipped that. That's on me. And that would be only specific to things that we think contain sperm. That would be differential wash would be useful for that. And that'll help minimize our problems with DNA mixtures downstream, like I was talking about earlier. But yeah, we're gonna get it, identify what it is, clean up the DNA, tell how much DNA there is and what kind of shape it's in, and then decide to go forward. And then the standard would be to go to the STR testing. And then if that goes well, then we can upload it into CODIS, whichever level of CODIS, and then hopefully get a hit. So I guess that would be kind of that's the ideal scenario, is like you upload it to CODIS, boom, hit, we're done, let the investigator know, and then they can go pick this guy up. It doesn't always go that way, but
right. But let's again assume that it's all gone that way. We've made it to the point where the profile has been, or the suspect cannot be excluded. And that has become part of a larger case for investigators, building against this suspect. An arrest is made, charges are filed, we go to trial. What role does a person in a lab, a scientist, a person who's worked on this evidence, what role does that person play in the trial phase of the process?
So yeah, that's a great question. So the lab is going to report the results out to the investigator, and those will have written conclusions that say things like that this person cannot be excluded, or that kind of whatever their specific language is. And so say we got a hit in CODIS. This guy's DNA matches the DNA from the crime scene, and they go forward. The investigator goes forward with prosecution. The lab person or the lab supervisor, depending on how they're organized, will often be called into court to testify to that evidence. And so what they're going to do is explain the evidence to the jury. And what does this mean? So if it was a mixture, there will be statistics that say, well, even if it's not a mixture, you'll say there are statistics like it is 1 septillion times more likely that the DNA came from this person than from another unrelated individual. And so we'll have these kind of statistics that say, how likely is this? We'll have like a likelihood ratio is one way to do it. And so they'll go in and they'll explain kind of the DNA evidence. And they're not there to say he did it. The lab person is there to say this, this is what the DNA evidence said and nothing else. They're not there to convict somebody. They're not there to exonerate anybody. They are there to just speak to what they tested in the lab. And I think that gets maybe frustrating for some people. And you have to be really careful if you're the person testifying for the DNA evidence to not overreach. because those other conclusions about who did a crime or that kind of stuff is going to be up to the, between the prosecutor and the defense. The lab person is just saying, I got this profile from this item, and it is the same or not the same, or here's the probability that it was the same if it's hard to tell, as this other profile that came from, this person. So I think it's more limited than you think, because I think in TV it makes for a way better drama if they're like, he did it.
It was that
Welcome,
one.
Dano,
Right,
So,
right.
but yeah, they're just there to testify to the evidence in terms of a lot of the interpretation of that evidence. That person's DNA was there. Or if they retrieved sperm from an intimate swab, that person's sperm was there. So
Right.
they're going to testify to that, and then it's going to be up to kind of the rest of the legal system to say how it got there, I guess. Would
Right.
be sort of
Right.
the long circuitous point that I just made.
Yeah, and a lot of times you see the defense, come to the table with witnesses who will refute or try to challenge the interpretations of the
Yeah.
lab that, couldn't exclude the defendant. Have you ever personally been in a scenario where your lab's findings, have been challenged by defense? And, is that frustrating as someone who believes in the work that they do?
I think, again, like the folks that work in the lab try really hard for a lack of bias. Obviously bias exists, but yeah, I mean, yeah, for sure. If you had worked a case and the DNA evidence was pretty clear cut, and then it's some kind of, like, maybe you felt like it was a technicality or how this person didn't get convicted. I'm sure that is frustrating, of course, but there's I don't know how to answer this. I think there's a fair amount of mental strain that's associated with folks that do this kind of work. It's just hard work. It's very exacting. It requires a lot of attention and diligence, and it doesn't always get a result at all. I think probably one of the most frustrating things is uploading a profile into Endus and no hit. No match to any other crime scene, no match to any known individual, nothing. And you get nothing back. You did everything right and you get nothing. I think that's frustrating. I think that's probably more common than like an epic defense trial showdown where somebody gets off on something that you thought was baloney. But that does happen. And yeah, of course it's frustrating. I mean, the people that work in labs and do this DNA work have dedicated their lives for the most part to getting it right. And so they want to see justice served. And I know, especially for folks looking from the outside into the lab, I think they're like, why don't they just do this? But it's far more constrained inside than out, because again, like I was talking about interpretation guidelines, you have to follow those all the time and not just sometimes. You can't test a kind of a thing that you have no experience testing, you don't have any protocol for. There are reasons why you can or can't do stuff. Some labs don't process bones at all, so skeletal remains they have to send out. It just sort of, it depends from lab to lab. But yeah, I think the testifying in court can be stressful. Mostly people are just really trying to get the evidence right, to get the stats right, to make sure that they don't say anything that's outside of their purview, to only speak to that evidence and not to things that are kind of ancillary to it that really ought to be left up to the prosecution. So it's stressful. I mean,
Well, I can imagine.
a lot of folks get really good at it, but yeah, it's not an easy thing.
Well, beyond the testifying bit, I imagine as DNA comes even more to the forefront, and we hear, left and right about cases being solved or closed thanks to DNA testing. With that, the misunderstanding that this is possible in every case becomes pressure on you and the people in the lab to, well, why don't you just test the DNA? So, I mean, I hope it goes without saying that I have a tremendous amount of respect for the work that you do and everybody else in the lab has done and has, carried that pressure with the increased microscope that's on your work.
Yeah, I mean, I think one of the things that is very different for forensic DNA analysts than pretty much almost any career I've ever seen. Imagine your work, whatever it is, on a daily basis, whatever it is that you normally do. You have to take such good notes on everything that you do, and you have to do it according to your protocols, because no matter what it is that you do, you can and probably will be called into court to testify to what you did on a specific day 2 years ago or whenever it was. And you have to be able to concisely explain what you did, to have such good notes that everything's in order, to obviously you study the case again before you go testify to it. But it's also the only field I've seen that if a DNA analyst goes to court to testify to evidence that they generated and they get disqualified as an expert witness, they're kind of done. You're good luck if you have to go. I mean, your lab is probably not going to send you back to court. And if you can't testify, then there's a lot of work you can't do. And so your career could end because you got disqualified in court one time.
Wow.
And so, there's definitely, I think that makes it hard to bring the technology forward in some cases, because if your job hinged on you being able to understand and explain these results, otherwise you might get disqualified as an expert and lose your job. You wouldn't trust a new technology until you understood it so well that you felt like you could do that, any day of the week without worrying about it. So I think that's, if we're talking bigger picture, if we're talking like, how come forensics is still doing this when they could be doing that? I think a lot of that reticence comes from the way that our court system is, this kind of adversarial system where these people's jobs are kind of on the line every time they have to go testify. And so they're not going to testify to anything that they don't feel comfortable with. And so they're not going to do anything that they don't feel comfortable testifying to. So anyway.
Wow. I mean, there's so much to consider. I never even thought of that aspect of it. So thank you. I want to make sure before we wrap up this intro topic into this series, at risk of opening a can of worms that would probably require its very own segment, where does the process change, or how does it change, if at all, if you're looking at evidence that's been in a storage locker for 20, 30 years, like talking about cold cases? Is it generally the same?
A lot of it is the same. The main things that are going to be different is that evidence ages not like fine wine. DNA is more persistent than you would think. And so if it's been stored properly, it's going to last pretty well. But, if you're going back to these cold cases, I think a couple of things to keep in mind are, like, if you had a fresh crime scene and you were like, you know what, we could really use another piece of this, or can you see if there was more blood spatter here, or that kind of thing. Well, if you're looking at a 30-year-old cold case, what's in that evidence room? That's it. Whatever's in that envelope, that box, whatever it is, that's all you have. There isn't anymore. But I think especially things that were worked in the kind of earlier days of DNA, when you needed a lot more DNA to get a result, I think it's exciting to be able to go back with newer technology and say, hey, look, we have a fair amount of DNA here, and we can do a good job, and we can get a profile from something that we couldn't before. I think it's frustrating sometimes when, and this happens occasionally, where people tried to work it as a cold case already, but maybe 15 years ago.
Mhm.
And so you're like, oh, okay, yeah, there's supposed to be swabs in here, and there's just sticks. Everything's been cut off. There's not really much left.
Mm.
But yeah, other than just those kind of little vagaries and the fact that the DNA has gotten older over time, but definitely the technology's gotten better. I think it's a lot the same. You're just hoping that that you can, find closure for these folks, figure out what happened.
Do you have any parting words or thoughts that you want to make sure people know about the timeline of DNA collection to results?
I think, yeah, I think I know it's frustrating. Um, and I've seen big changes happen in forensics since I've been a part of it, where, I mean, 10 years ago, 10 years ago, probably, people started looking at untested sexual assault kits. And there were warehouses full of these things. They'd never been tested. That, I mean, you don't have to be educated in anything to know that that's a problem. Like, that's not okay. But I saw several states that mandated X, Y, and Z in terms of sexual assault kit testing, and then had legislation that said you have to do it. You have this many days to turn it around. You have all this, and then no money to do it with. So I would say parting words. I mean, if you have your state and federal representatives, if you see things like the Debbie Smith Act coming up for a vote, or Violence Against Women Act, or the Sexual Assault Kit Initiative, or things like that, write them a letter, say, vote for this stuff. This is important. Provide money to back this up. We want to do it. We want to do it all, but it's not free. I mean, you have to pay for it somehow. So I would say, get involved. Don't just say, yeah, you got to test that. You figure out how to pay for it too. There's we would love to, but I think a lot of groups that did happen for the sexual assault kit testing. The Psaki initiative was huge. Debbie Smith Act helped a lot. The Violence Against Women Act helped a ton. Lots of grant funds. The National Institute of Justice provided a lot of grants and money to labs to test their own samples, and also to outsource them to private labs if they didn't have enough people to work them all. And by and large, those sexual assault kit backlogs have been, if in a lot of places, have been completely erased. There is no backlog anymore. And in other places, they've taken it way down. And so things are being processed a lot faster. And in some cases, things are just being processed. I mean, New York was one of the first ones to do it, and they didn't even try to triage. They called it the forklift approach. Just whatever fits on the forklift closest to the door, okay, pick that up, and then we're going to take it to the lab and we're going to test all those. I think the interesting thing, just side note from all of that, is because they didn't look at the cases or attempt to triage it in any way, a lot of kids were never tested because it was acquaintance rape. Those profiles hit to all kinds of unsolved stranger sexual assaults, because it turns out if you're enough of a jerk face to do that to somebody that you may also do that to a stranger.
Right.
So the evidence that, I mean, COP was like, well, we know who it is. There's no point in testing that. Maybe there is a point. So I think that kind of, we're all learning. DNA is still fairly new, not really, but in terms of all technology, of all science, we didn't have the machines to make copies of DNA in the lab until after I was already in school, in like university. So it's not,
Say when that
I mean, I'm
is,
old,
like,
but I'm not that old. Like, this stuff has come about pretty quickly, and it's growing in leaps and bounds. I think the other thing I would say is that, as frustrating it is to not get a hit in CODIS or ENDIS or whatever, there are more things we can do. And genetic genealogy obviously has been blowing up since the Golden State Killer case. And I would like to see, we talk about funding, we talk about having to testify to results, but there is no major reason why your city's, county, or state crime lab could not do DNA sequencing on a level that would be enough to get a genetic genealogy profile. I would like to see it where if you don't get a hit in CODIS and you have enough DNA, automatically go to genetic genealogy and see if you can identify that person. But we democratize sequencing. It shouldn't just be in the hands of a few people. It ought to be in everybody's hands. So hopefully that'll happen.
Well, this has been an absolutely illuminating conversation. Thank you for
feel
leading me
like-
through it.
Hopefully
truly-
nobody's died of boredom out there. If you are, please splash some cold water in your face. It's gonna be fine.
Refresh. No, I'm sure everyone is feeling just as educated and lit up by this stuff. Even if we have lingering questions, there's that.
I hope
means
we do.
room for more exploration. Yeah, absolutely. So thank you, Meredith. And until
Sure.
next time, the next topic, we'll see you then.